I'm here at ISHI 34, and I'm happy to be joined with Brian Renell from Diacel Arbor Biosciences. How are you?
Good today. How are you?
I'm good. Thank you for asking. So, off camera, you told me that this is your company's first time at ISHI.
That is
Yes.
correct. Yep.
So please tell me a little bit about how you all got the chance to come here.
So we've made products that have been helping the forensic community for several years, but we've never really reached out to the community to learn more about what applications they're doing, where are their pain points, and where are opportunities for us to help the community. And so I'm here to learn.
Yeah. And ISHI is the perfect place to do that.
That's what we've heard.
So tell me a little bit more background about the company itself, how it got its start.
So we were started by two professors from the University of Michigan, and we're still located in Ann Arbor, Michigan. We started about 2008. Originally, we were making microarrays, and we've then adapted that technology and other technologies to create a new platform called Hybridization Capture. So we have probes for enrichment of DNA. So this is where it's really powerful in a community like the forensic community, where they work with very degraded, contaminated samples. We can create specific probes that allow you to enrich specific regions. So it's like finding the needle in a haystack. Yeah. So instead of sequencing your entire sample, you only sequence a region of interest, and it's a very powerful and cost-efficient process.
Nice. Okay. So what other services do you all provide that could be beneficial to the forensic community?
So we provide custom solutions. So if someone has a specific thing they want to look at, I can help them and create a custom tool for them. We also provide services. We can either send the kit to the customer to use, or the customer can send us our DNA and we do all the work for them.
Okay.
So we offer lots of flexibility and options for whatever the customer really needs for their research goals, their project, and their budget.
Nice. So custom solutions, can you give an example of that?
Sure. So someone has 10,000 SNPs in a genome they want to study. I can make them probes that instead of sequencing the entire genome, they only pick out those 10,000 regions from the genome for study. So instead of sequencing everything, you sequence a very small portion of it.
Right.
Really powerful if that sample came from something that isn't contaminated with environmental bacteria.
Right.
That allows you to, again, really just focus your energy and your money on only the regions that are important to you for your analysis.
Okay. And you said you all have worked with degraded sample types?
Correct.
Go ahead.
I'd say we also have worked very much with the ancient DNA community. So we've done enrichment on mammoth, sabertooth tiger, 4,000-year-old mummies.
Yeah, contributing to history.
And yeah, ancient DNA, human remains, for us, they're very similar. We know that how important they are, how rare they are, and how difficult they can be to work with.
Yeah, well, that's perfect, because I was going to ask you what are some of those degraded sample types that you have worked with. So ancient bone DNA.
Yeah, ancient viruses, ancient bacteria. And we work with modern things too. We're not just the poor sample people.
Right.
We can work with everything, but our strengths and our experience and our background are all really just with what we like to call problematic samples.
Nice. Okay, and have you worked with any of these personally?
I do more of the bioinformatics and the customer support.
Okay.
So I have done study, or I've helped people with their studies in order to then look at some of the ancient tuberculosis from mummies type of thing.
Ancient tuberculosis from mummies.
Yes.
Oh my gosh.
And Epstein-Barr virus from mummies. So there's a lot of, and mummies doesn't necessarily mean Egypt. It just means, mummified remains that could be, very old and maybe only hundreds of years old, but sometimes thousands of years old.
So, okay, this is just me not knowing. So let's say these ancient viruses from mummified remains, where exactly do you get it from within the remains?
Well, that's why target enrichment is really good. You take the entire sample, and instead of trying to sequence all of it, which is mostly going to be the environment and then the human, we specifically enrich for the virus sequences. So instead of sequencing everything in that sample, we pull out the regions that you want.
So when you say the sample, what is the sample?
The sample could be like a scraping or a swab.
Okay. Wow. So tell me more about your role at Diacel Arbor Biosciences.
I'm the Cedar product manager for MyBaits, which is the product that allows people to do the targeted enrichment. And so I work with customers, find out what their needs are, and then I do the bioinformatics to create the probes for them that will hopefully answer their questions. Okay. So we very much base our communications on trying to figure out what their needs are and then addressing those needs.
Nice.
So I get to talk to a lot of scientists and I get to learn a lot as well as help them.
So biosciences, you said working with the codes, you develop the code, do you then go out to teach them how to use the code? Or you do all the code and just send them?
I do all the code and then I give them the product.
Okay, right. So you do all those heavy lifting.
Yeah. Some customers are very adept and probably even better than me and do it, but there's a lot of people who know, like, this is my endpoint, how do I get there? And we help them through that series of steps in order to get to their goals.
Nice. So this is also your first time at ISHI?
Correct.
Yes. How have you been liking it so far?
I've been enjoying the talks, meeting the people, learning a lot of, I've worked for the federal government for 12 years, and so it's no surprise that apparently there's a lot of regulations and other aspects that take a long time for some of the new technology to be accepted. And so I'm learning more about what those next steps will be, because I'm really curious about, the next level of whole genome sequencing and how it could be used for forensic analysis. It seems like there are gold standards, and then there's, things that are accepted technology but haven't been accepted necessarily universally in the court systems or different administrations.
Right.
But I'd say that's what I'm learning a lot is like, what are the holdups in a way? Not holdups, but like barriers.
Right. Yeah.
Limitations or slow, yeah.
So what's something that has shocked you, or I guess something that surprised you that you've learned from being here at ISHI?
So I've been doing a lot of next generation sequencing for years and years. It's, the technology came out a while ago. It still hasn't seemed to be implemented, and a lot, across everyone. A lot of people I talked to said they're not doing it yet. So I believe they know that is the next step for some of the, this research and application, but they haven't gotten there yet. Right. And, learning that, where their needs are, where they are, and getting there has been very important. But I would say, coming from a world where we sequence everything to a world where they know that's a good idea, but haven't necessarily implemented it across, the board was not expected.
Okay, it's interesting. So have you been able to explore Denver?
Not that much yet. I have been in Denver in the past, and Estes Park and Rocky National Park, and this is a beautiful place.
Yeah, it is. So is there anything else you would like us to know about Arbor Biosciences?
Probably should say something. I work with a great group of scientists. We love working with people. We love challenging problems because we like to think, we can help and create solutions and everyone wins in that sense.
So what's the most challenging problem you have come across?
So there's different problems along the way, but for me, one of the challenging problems is we can make a probe to enrich anything, but if let's say that region occurs multiple times in a genome, then it's not specific. It's trying to figure out the best way to enrich a challenging target. This doesn't probably make too much sense out loud, but it made sense in my head.
Yeah. Well, thank you so much for speaking with me.
Thank you.
And I hope you enjoy the rest of ISHI.
Thank you very much.